All right, welcome everybody. I'm Ben Brewer. I'm here to introduce Dr. Tim Sanis. Let me give you a little bit of background on our presenter today. So Tim has an undergraduate degree in psychology from UNC Chapel Hill. Following his graduation, he spent 2 years at the NIH working with NCAM and studying mind-body medicine. He earned his PhD at University of Florida's health psychology program, and then did his clinical internship at the Puget Sound VA. He then entered into our T32 fellowship under the direction of Dr. Gene Kuttner in palliative care and aging, and during that time worked closely with Mark Laudenschlager in the behavioral immunology and endocrinology lab, where he made significant contributions to our Corey-funded study involving the caregivers of allogeneic stem cell transplant recipients. Following his fellowship, we hired Tim as an assistant professor in the Division of Hematology, where he works currently with patients and caregivers in the BMT program, as well as co-facilitating the caregiver support group for the Cancer Center. He continues to be very productive in the field of psychoneuroimmunology, with a research focus on the biobehavioral impacts of the caregiving role and shared decision-making, as well as considerable amount of work with cortisol. So please join me in welcoming Dr. Tim Sanis.
Thanks, Ben, for that lovely introduction. I'm going to keep an eye on time here. Okay. This is a really long title, so thank you for all for at least piquing your interest a little bit, enough for you to come here. Maybe it was the food, but I do want to have a little caveat that I'll talk about this exact topic and one approach that we've taken to combining telomere length and telomerase. However, I will talk a little bit about some other stress marker psychoneuroimmunology work that I've completed while I've been here at University of Colorado. So I have no disclosures, and I stole this slide from American Psychosomatic Society, where I presented this work in a much more abbreviated form last week in Vancouver. So I'm glad to be back here, but that was a pretty beautiful place. So I left this, that's the picture up there. So no disclosures. So just by way of overview, so I'll talk about where Dr. Brewer and I live, which is in hematopoietic stem cell transplant, how there's a need for caregivers for any patients undergoing that transplant. I'll talk about associated stress in those caregivers. And then I'll talk about measuring physiological stress and some approaches that we've taken to doing that. And that caregiving, what do you know, is associated with some negative health outcomes. Then I'll talk again about the topic at hand, which is telomeres, and in this, what I view as an innovative approach to combining telomerase activity with telomere length. And then finally, I'll talk about a little maybe tangential idea that I have, but how this might apply to all of us, to consumers of kind of emerging biomarker information, which I think is an important direction. So, as I mentioned, I hope my animations come up, because this doesn't bode well. But anyway, this is a typical time course of hematopoietic stem cell transplantation. This says it's for autologous stem cell transplant, so that's where you take your own cells and they're infused back into your body for treatment of hematologic malignancies. However, the recovery and the timeline is lengthy. That's really the point of this slide, is to show you that we select folks that have, again, these life-threatening hematologic malignancies for transplant, and it's not just selection based on their disease. Right. This is again a long process. So that on the gray bar there, it's up to 100 days, typically post-transplant, that we require patients and their caregivers to stay locally, so that we can closely monitor the various things and complications that can come from this intense procedure. So they're typically in the hospital for a month for the allogeneic stem cell transplant. And again, that recovery then is a very slow process, and we need to closely monitor them. So it really uproots folks from their lives, and the caregiving piece is, at least in our program, is a requirement that they have a family member, a friend who's able to volunteer for this, essentially unpaid responsibility for 24/7 care. care. So again, it's a lengthy, difficult process and I mentioned that these caregivers are available for 100 days. And what do you know? These caregivers are particularly stressed. So if you compare them to patients, oftentimes they actually have more anxiety, more depression, more stress than the patients for whom they care. And so multiple reviews have essentially showed that caregiving in this context of hematopoietic stem cell transplant is burdensome and disruptive to caregivers' lives. And yet, again, a requirement for, at least in our program, for undertaking this. So that's just a really brief overview about the context in which I practice clinically, but also how we view this as essentially a model for studying caregiving stress. So I'll talk about, again, not related to the title of the talk, but it's something I've become very interested in during my postdoctoral fellowship, is how we measure cortisol. So for those that aren't familiar, this is a nice review by Mike and Tony in Nature Reviews Cancer back in 2006, basically just outlining the hypothalamic pituitary adrenal axis. And stress is this kind of, it's often these like lightning bolts at the top of the brain, because they don't spend much time talking about what is actually interpreted as stress. But the point of this slide is that as this cascade begins, when your body, your brain identifies a threat, that the cascade essentially goes through the CRF, the autonomic nervous system releasing adrenaline, and this neuroendocrine response, and essentially cortisol is something at the end that we often find we can measure pretty easily, okay? So you can measure this a number of different ways, but it's certainly detectable in saliva. So salivary cortisol is fairly easy to measure. It's fairly easy to collect. A little bit burdensome for participants. We often have to get it throughout the course of the day, and I'll talk about that. But some weaknesses that have been identified and where I really became interested is in whether or not this is actually sensitive to self-report. So the subjective experience that folks go through, in this case caregivers, and whether or not it's actually sensitive to change. So I won't talk about the work I've done in sensitivity to change, but this idea that it may be related to again, self-report I think is an important direction. So I'll try not to bore you with statistics in this talk, but I will have to kind of delve in a little bit to some of the work that I've done in that area. Essentially, cortisol is even though it might be easy to measure in saliva, and I'll talk about some other ways towards the end, it's quite difficult to quantify the rhythm. So there's been some interest in this idea that's called intra-individual variability in the cortisol response or production throughout the day. And that's essentially what I'll show you here graphically in a second. But there's some emerging data that those with major depressive disorder, those with remitted bipolar disorder have this erratic kind of cortisol output. So what does that look like? So if you have cortisol on the y-axis here, in time of day, here on the bottom, you basically can summarize through a given participant collecting these salivary cortisol samples throughout the day. You can essentially summarize it with a line, okay? We call that the cortisol slope. And anyone, that's a pretty basic statistical approach. You maybe collect it for 3 days. In what I'll present, it's about 4 times per day. So you aggregate across those 3 days and create this kind of average slope for an individual. That's been shown to be related, again, to major depressive disorder, a kind of flattening of the slope. But if you look at the actual data, what it looks like is something like this. So the squiggly line didn't match up, but the figure came out sort of. That there's a lot of variability around an individual's slope, okay? And so what we aim to do is really essentially capture that variability. And others have done this, and this is building on work by Rachel Yehuda at Mount Sinai. Early on in the 90s actually showed that this more advanced statistics can pick up on, it can actually distinguish between those with post-traumatic stress disorder and those with major depression. So what that looks like is if you see a depressed individual on the here on the right, and this is repeated sampling. So this is much more intense than what we ended up doing, but this is an indwelling catheter that essentially, a PICC line that essentially captures cortisol over in 15-minute epochs, that this is much more variable than the participant on the left, who essentially is not depressed. It has a much more normal, less erratic cortisol rhythm. And in PTSD, it turns out that this is actually related to a more consistent cortisol output. So the kind of flip side of being depressed in that erratic cortisol output, those with more anxiety disorders seem to have this very tightly regulated cortisol rhythm throughout the day. So we extended, this has also been extended to in very, very even more sophisticated statistical techniques. They've replicated these findings. And again, this is shown here with the non-depressed, non-PTSD participant on the top, that with the erratic output I mentioned in the middle, and then in PTSD on the bottom. So you can see it's very tightly regulated. So again, my interest is in whether or not these cortisol dynamics were related to self-report measures. And I started with talking about caregivers and hematopoietic stem cell transplant, so that's essentially where we took this. So we looked at this in again, saliva samples throughout the day, and caregivers approaching their caregiving responsibilities for the stem cell transplant, and showed that indeed those with this kind of composite score, combining a number of self-report measures, those with more mental health, high mental health is more distress. On the right, those were a more erratic rhythm as they approached their caregiving responsibilities. Those that were seen to be doing okay on the left, they really didn't have much of this higher inter-individual variability in cortisol. So we published this in Psychosomatic Medicine. And I will say, and this is one of the only ideas I've ever had that actually has gotten a little traction, a lot of folks have actually extended this to other data sets because there's a lot of folks that have collected these salivary cortisol samples and, essentially applied other types of analyses like slope that I mentioned. But now it appears this variability estimate is somewhat meaningful. So that was again a lot of the work that I did in postdoc, looking at these variability rhythms, and whether or not they're meaningful in larger studies, whether or not we can really make sense of them, again, is an ongoing question. But I want to talk just a little bit about why we think measuring these markers in caregivers is particularly important. So this is a whole area of research, and these three kind of studies that I highlight on this slide really don't do it justice. But Richard Schultz in 1999 was able to show in a fairly large study, about 300 folks published in JAMA, that those providing care, this was Alzheimer's patients for whom these caregivers were providing care, actually had a 63% increased mortality risk compared to non-caregiving controls. And it seemed to be related to those that not only were caregiving, but those that also expressed strain with their caregiving role, okay? And so there's been some follow-up studies, and I mentioned Vitaliano in 2004 with a meta-analysis suggesting that cardiovascular disease, there's some suggestion for increased risk for cancer, and a number of other different health outcomes were related to this role of caregiving. And it should be noted that in that meta-analysis, that stress hormones showed some of the strongest relationships with this caregiving role. So there's been a lot of work in this area. It also should be noted that those participants that they looked at in the meta-analyses, their own individual interpretation of their health seemed to be related to this role too. So potentially caregivers' self-perception of their health, but then also again, these biological markers and the overall kind of health risk of the caregiving role. They've also drilled down into actual incidence of hypertension and shown that 8 years post kind of taking on a caregiving role, that caregivers had a higher risk for hypertension. And so again, there's a lot of epidemiological and broad evidence that caregiving at least confers a little bit of health risk for negative outcomes. I didn't highlight, there's a little bit of evidence coming out in the last few years, potentially to the contrary, where maybe there is actually some health, not necessarily benefit, but the folks actually get a positive psychological experience from their caregiving role. But this is all by way of background that caregiving matters and is related to these important endpoints. So now switching gears in the topic at hand. So there's a lot of folks in this room that know measurement of telomeres probably better than myself, and maybe some of the background and historical relevance of measuring telomeres. But essentially, measuring telomeres was initially discovered by Elizabeth Blackburn in Gaul in the late 70s. And these are the ends of chromosomes. So they're not really well highlighted in that green chromosome figure, but these caps protect from deterioration and fusion with neighboring chromosomes. So really important that every time the cell divides, that these telomeres protect the cell and then actually shorten in each division. So that in the scientific literature, but certainly in the pop culture, that these have been really kind of referenced as a cellular marker of aging. And so it's gotten a lot of traction in a number of different fields, and certainly in what I'll talk about with stress biology. I'm also going to talk a little bit about telomerase, and so this is the enzyme. This was discovered by Elizabeth Blackburn and Greeter in 1984, and this is the repair enzyme that is species dependent, and it's a region of repetitive sequence at each end of the chromosomes. And so, again, this is, for those of you who aren't familiar with this area, Elizabeth Blackburn, Greeter, and Stozak, they were awarded the Nobel Prize in 2009 for not only the telomere length discovery, but then also this repair enzyme, and the importance of that in not only aging, but in just cellular division, cancer. It's been applied to a lot of different areas. And so I won't go through this because, again, this is there's a lot of trust in the laboratory techniques that Mark Laudenslager uses and collaborates with the Blackburn lab. But this is essentially a diagram of how telomerase works through the flanking end of the telomeres and to actually repair the lagging strands of the telomere through that process. So again, those that have probably read popular science and those kinds of things, there's been a lot of interest in, okay, if this is a cellular marker of aging, how is this related to folks' well-being and how they're doing psychologically? And so I'll talk about Alyssa Eppel's work. She's been a pioneer in this field, and that's who we collaborate on this project. But I should say that if you look at the more recent literature on markers of well-being, self-report measures of how people are doing, and this telomere length or telomerase, that actually it's a little bit of a mixed story. So I just pulled a couple of the larger studies looking at telomere length. And so in the first study, 2016, that again, the larger cohorts studies that exist, so remitted and current depressive or anxiety disorder has shorter telomeres compared with healthy controls, so just group comparison. But they're actually over the 6 years measured in the study, no changes in those depressive, anxious symptoms were related to telomere length. And so whether or not sensitive to change, I think, is an open question. Jonathan Schaefer, who's actually at the downtown campus now, he analyzed a very similar Nova Scotia health study. It was a huge number of participants and wasn't able to find a relationship between telomere length and depressive symptoms. And more recently, or I shouldn't say more recently, but certainly one of the more recent meta-analyses of 20 different studies looking at this question really showed a very small pooled effect size between telomere length and perceived stress, which is one of the more common psychological measures of how people are feeling, typically in the last 30 days, but just this measure of again, that internal experience of stress. So I just want to, again, walk through a couple of studies that I think are really exemplify what our current thinking and I think leaders in the field view as important to this idea of psychological stress and telomere length. So in this study by Surtees, it's in 2011, they were actually able to show that negative experiences in childhood, and this was things like being abandoned, being exposed to alcoholic parents, and that summary of those difficult experiences was indeed related to telomere length. So that was in that study, they were not related to these self-report measures of how people were doing currently. But if they tallied up how they were doing and exposed to these negative experiences in childhood, that was related. And very similarly, Wolkowitz, who's, again, you see a San Francisco and a leader in this field, in conjunction with Elizabeth Blackburn, similarly showed that current depressive symptoms, not related to telomere length, but the chronicity, the lifetime duration of depression, was related. So that how many times these individuals with major depression had gone through depressive episodes, that seemed to have a cumulative effect on the telomere length. So it's more of the suggestion of early life experiences, cumulative life experience being related to this important marker, and not necessarily these current subjective states. So I'm going to try to bring some of that together and talk about the little bit of work that's been done in telomeres and caregiving. So I mentioned Alyssa Eppel, who collaborates with us on this project. She continues to work closely with Elizabeth Blackburn, looking at these topics. But essentially, the first study was that she worked on in this area was comparing mothers of healthy children with those of mothers with a chronically ill child, and showed that, and I'll walk through the actual data here in a second, that shorter telomere length was related to this chronicity, the number of years caregiving, and actually in this study it was related to perceived stress. Interestingly, the high stress group, when you kind of broke them out into quartiles, had lower telomerase activity. So a little bit, maybe not exactly what we would anticipate. And then there's a 2007 study showed that caregivers, when compared to matched controls, and this is in Alzheimer's, caregivers had significantly shorter telomere length overall. And the telomerase activity in that study was actually higher in the controls. So teasing apart these dynamics, telomere length, telomerase, it's not quite a clear, consistent story. But this is just the actual figure from Alyssa Eppel's work, showing this chronicity in years on the top of caregiving and its relationship to telomere length. And again, what I've not seen as much in the literature since this study, but the perceived stress scale showing this kind of modest effect with telomere length. So this is a Jan Kiecolt Glaser, who's the National Academy of Sciences, is basically a pioneer in this general area, did some of the early work. She's at Ohio State, and she did a lot of this early work in childhood adversity associated with short telomeres in caregivers. So she had this really elegant paper showing that there's this kind of messed up and not presented, but you can see that there's this, in looking at childhood adversity, and she used something called the ACEs, so Adverse Childhood Experiences Survey, that there's this stepwise progression of more childhood adversity. On the y-axis, there's actually IL-6, a pro-inflammatory cytokine, in a very similar relationship to those that experienced abuse versus those that had not. And then in the almost identical relationship in a stepwise fashion to having no adverse childhood experiences versus kind of endorsing one versus multiple, and showing that kind of stepwise progression of lower telomere length. Interestingly, because this was a group of caregivers and controls, there was actually an interaction where there was a kind of a double hit. If you had a history of negative childhood experiences, abuse, and you're in this caregiving role. So that had even more impact on the inflammatory cytokine in this graph, TNF alpha. So when I actually took an initial stab at this work, or this data set, I should say, we really showed a very modest relationship between telomere length and anxiety. So it wasn't related to depression, it wasn't related to other reports of well-being, but there seemed to be something going on with the duration that caregivers had actually been in this role. So that got us thinking more about how we could tease this out. So now I'll actually get to the goods of why you guys came here and combining these telomerase activity with telomere length. So, sorry, some of my figures are not showing up. That's okay. So basically, Zali and colleagues in Presidium National Academy of Sciences had this really elegant paper, or at least I thought it was interesting, not necessarily in caregivers, but what they argued is that looking at telomere length or telomerase individually is at a cross-sectional time point is probably not that meaningful. What actually is potentially more meaningful is, again, these dynamics of telomere length and telomerase activity. So what they did is they had this median split between those with long telomeres. So we don't know what really what's going on there, but we're actually not as interested because at baseline, they don't seem to have any of this telomere shortening. But then within the telomere, those with the shorter telomeres in the median split, they had those with high telomerase activity and those with low telomerase activity. So then it creates these three groups. And so what they're able to show, and again, just to review, the stress phenotype would be the high telomerase, long telomere length, and or the short telomere length and the high telomerase activity. So what they're able to show is that indeed these, when you've put someone through a stressful laboratory tasks, as anyone hear the Trier social stress test? It's kind of like what I'm doing now. It's like you stand in front of a judging audience and they kind of look at you weird, and then they measure your stress response. Anyway, it's like very validated as one of the most stressful things that you can do. So, but they're really like harsh critics. So that's what they put these folks through. Again, not caregivers, but this division of median split between these three groups showed a very, very clear pattern of a blunted in the stress phenotype, the short telomere length and the high telomerase activity. They showed a blunted response to this stressor, suggesting that something's going on different physiologically, and that was both in heart rate variability and systolic blood pressure recovery from that task. They also showed that these three groups that I mentioned, so that they're really outlined there, had significant differences in optimism, social support, and in hostility, and in the predicted directions, actually. So that in this sample, there was actually a significant relationship between the groups on these self-report measures that it seemed, in the prior work that I had mentioned, did not show significant relationships but with telomere length by itself or just telomerase activity. So just to summarize all that background, so I initially was talking about how some of my initial interest in cortisol variability and how that might be related to this caregiving experience. We've moved on to telomere analysis, and it seems that that this might be a worthwhile direction in terms of prior experience seems to be related to telomere length, more so than these individuals' reports of well-being. So maybe it is a cumulative marker of stress, and that this combination pioneered by Zali and colleagues may tease out some of that. So on to the current study. So these participants were recruited, they were caregivers. I started with the idea of hematopoietic stem cell transplant, and so these are caregivers that were had volunteered not only to serve in this caregiving role, but also for the study. And it really is part of our routine evaluation. So this is a study, as Ben mentioned, was funded by Patient-Centered Outcomes Research Institute, or PCORI, and that our clinical process requires us to kind of not only recruit the caregiver, but also kind of vet them and make sure that they're prepared for this role. I should report that this study was actually a stress management intervention, and then I'll only talk about the baseline data. So this is a cross-sectional analysis, and does not look at, even though it's an interesting future direction, we did not look at these telomerase activity dynamics over time. So I won't go, I already kind of mentioned that I did process some of these blood samples, but if you want to talk about how we actually measured telomeres and telomerase, we can do that offline. But essentially, the Blackburn Laboratory was our collaborator on this, and we isolated white blood cells, peripheral blood mononuclear cells specifically, and lysed them to measure telomerase, which is actually, for the laboratory folks in the audience, it's about an extra 3-hour process compared to telomere length, which is really just you can store the white blood cells and measure that at your convenience if they're kept frozen. So I've already mentioned that we were going to pursue this approach of the median split between long telomeres, short telomeres, and then another median split with telomerase activity. This results in 3 different groups. So then, how do you measure chronicity of caregiving? Okay, this wasn't our initial plan, and we didn't have the Adverse Childhood Experiences Survey that I mentioned. It's called ACEs. We didn't have a lot from what folks had been through throughout their lives, but we did know that many of these caregivers were new to this role. Okay, hematologic malignancies often come on unexpectedly. Caregivers might be thrust into this. Sometimes the symptoms may have been there for a year, years, but that oftentimes in terms of going through allogeneic stem cell transplant, the caregivers are kind of thrust into this role. So I wrapped my head around this for some time and thought about, okay, what other caregiving roles might these folks take on? And one of them, the question that we did ask is, have you ever been a caregiver in the past? Okay. The other one is, are you currently caregiving for anyone else? Do you have any dependent children living with you? Now we can debate whether that's another caregiving role, but those parents that in the room know that oftentimes the summation of all this stuff it can be stressful, to say the least. So we summed these 3 questions, thinking that again is a measure of combined caregiving roles. And so this is a 3-level variable used to predict the above group membership. So if anyone wants to talk about multinomial logistic regression, I'm happy to, but I'd rather not. It's really just a technique that you use to predict a 3-level a dependent variable if you have a number of different predictors and some covariates that you're interested in. So I mentioned the sum of other caregiving roles, and we also used some of this slide probably should be updated because I've extended these analyses in the last week, actually, in preparation for last week's talk. So we have the sum of these caregiving roles, and we have the length of the patient's illness. Okay, we initially chose that as kind of a proxy for how long these caregivers have been in this role, but then we also looked at, in addition to these 2 measures, how long they stated they had been caregiving. Okay, I mentioned the reference group, the 3 different groups that we're interested in, and in these types of these logistic regression models, you pick a comparison group, and in that sense, we wanted to compare the stressed phenotype, which is this short telomere length and high telomerase activity, to the other groups. And then as kind of more exploratory, because I already made the case that these self-report measures of well-being might not be as meaningful in the story of telomere length and telomerase, but we wanted to at least explore whether or not these self-report measures, specifically combined distress, burden, which is a kind of validated measure of caregiver burden, and then sleep were related to these 3 group memberships. We tested a whole bunch of covariates that I could talk more about, but a priori, we know that age is related to telomere length. That's, again, the crux of that measurement, and that there's often sex differences. So a priori, we kept those in the model. So this is 143 caregivers providing full data. For better or worse, it's a fairly homogeneous sample, primarily Caucasian females. They're non-Hispanic. They're incredibly healthy caregivers, and I don't know if they're lying to us, but they don't. Almost none of them smoke. Most of them had at least a few years of college, and the majority were spouses of the patients for whom they care. Now, I did mention this median split of these 3 different groups, and so there's the breakdown of how many folks ended up in each of the groups. And here's just the breakdown of many actually said had not been prior caregivers, 58%. The 92% were not caring for someone else, and 69% did not have dependent children living with them. So it gives you a sense that even though this is, I think, a valid question, a lot of these caregivers were fairly new to this role. 18 months was the average length of patient's illness, which if you compare that to the Alzheimer's caregiving literature, many of those folks have been caregiving for years. So again, it confirms kind of the hypothesis that a lot of these caregivers are fairly new and that they're quite focused on their patient oftentimes who is their spouse. So we confirmed that there's this negative relationship between, as age increases, length of telomeres goes down. We expected that. And then this is what I mentioned when we really looked at initial stab, looking at anxiety, depression, and caregiver reaction assessment to telomere length. Sorry. And basically only found that there was a modest, small relationship between anxiety and telomere length. And this is just a breakdown of that regression model. But this was the first indication that potentially this duration of illness might be related just to telomere length itself. And maybe there's a small relationship with anxiety. And telomerase activity was independently related to anxiety. But let me get to some of the more interesting stuff. So the reason I didn't initially go with caregiver self-reported history, so how long have you been doing this, is because it has this kind of weird distribution. So anybody that's taken basic stats knows this is like a skewed distribution. The majority of folks are saying they hadn't done this for very long. Actually, they hadn't done it for at all. They'd done probably like a month, something like that. So the way that I'm currently thinking about this is actually splitting this variable up into either quartiles or even a median split. But I will say that any way I run it, I get the same results I'm about to present. So basically, the summary of prior caregiving responsibilities and the length of patient's illness were both significantly related to membership into this short telomere length, high telomerase activity group. And that's in comparison to the other groups there listed. There's another, again, way that I looked at this, splitting up the self-reported caregiving duration, and that was also significantly related. However, when you look at the stress composite, sleep measurement, caregiver reaction assessment, these self-reports of well-being, there's almost nothing there. It does not distinguish between the three groups that we're interested in. And so this is shown graphically here with the patient's mean duration of illness on the y-axis, and then that you can see that the middle group, which we consider the stress phenotype, has significantly longer in terms of their patient's duration of illness. This is shown again here with the duration of illness in blue, and then the self-reported caregiver duration in red. And so it's actually even more dramatic when you look at it that way. And it's what I'm kind of leaning towards. And if anyone has feedback, I'd love it. The quartiles, when you break out caregivers' self-reported duration of caregiving, seems to really be the strongest relationship into these three, the membership into the middle group there. Across depression, across anxiety, there's really there's no differentiation between these three groups. So to summarize what I just presented, so it seems that the chronicity of caregiving is related to the stress phenotype of a short telomere length and a high telomerase activity, both in summary of other caregiving responsibilities and in the duration of their patient's illness. And it may be that this combination, pioneered by Zeli and colleagues, can tease out these unique relationships of caregiving chronicity. And I found it quite interesting that the self-report measures of well-being were not related to these group memberships, maybe a slight association with anxiety, but nothing to write home about, not a very strong relationship. So is this related to the literature of early adverse experiences? Potentially. Chronicity of caregiving could be considered one of those kind of hits to the system, so to speak, that has been shown in other telomere work. And I'm currently investigating this longitudinally. I mentioned this is a randomized clinical trial, and whether or not those three groups that I've highlighted could be related to response to intervention. So there's a number of limitations in the study. It's a homogeneous sample of caregivers. It's cross-sectional, so obviously there's no causality. We don't know what's really driving this, but it's certainly interesting. And then like any good statistician, I've continued to think about potential moderators, so new caregivers versus experienced, if they've received help from others. That's something we haven't really delved into. But I think that what I've tried to highlight is that this chronicity of caregiving is probably a pretty complicated story. So I just want to finish with a couple ideas that I have, and this is for us as consumers of information, those that are interested in pop science and where the kind of future is going in terms of consumerism and stress measurement. This is something I propose as a career development award for those that are interested. We can talk more about it, but I think there actually are a lot of implications for stress measurement. So I talked a bunch at the beginning about this cortisol variability, but they also highlighted some of the challenges in that. Future directions as far as that goes is measuring it much simpler measurement, right? And so Mark Laudenslager is a pioneer in measuring cortisol in hair. And the cool thing about that is you can just get one number, okay? And there's some meta-analyses suggesting that there's a relationship between a small relationship between perceived stress and hair cortisol. And it may be, again, that there's some relationship to life adversity. But it's still being worked out. So the reason I bring that up is not because I need to talk more about stress measurement in hair, but because you can actually order this online. So I don't get money from these companies. They probably would not like me talking about them this way, but this is commercially available. And what you'll get is these reports of how normal or abnormal your hair cortisol is. So I don't know what that means at all, and I would actually argue that we're not quite ready for that scientifically, but it's there, okay? And I think that one of the challenges potentially for many of us as scientists, but certainly in the kind of stress physiology world is what do we do with these numbers? Is it meaningful? Do we care? And if someone brings these types of tests into you as a provider, what do you do with it? And so that's... in that grant, I actually got a lot of guidance from like the ethics consult here and CCTSI in preparing that. But you can also do it with telomeres, okay? So you can, I have no, again, vested interest in this company, but you can help your cells stay younger longer. I don't really know if that's possible, but what they advertise is that they can give you not only your initial measurements, but your measurements over time. So that kind of brings up some unique questions about what people do with these data, how they interpret them, if they're amenable to change. So there's a lot of people writing about, is this snake oil? What does it mean? But we've actually, in the caregiver stress management study that I mentioned and was presenting baseline data on, we actually did do some biofeedback, and I'm interested in delving into that data in terms of how people interpret this, whether it increases their engagement with the intervention, because it does provide real-time feedback in terms of their heart rate variability. So a different stress measurement, but I think that there's a lot of overlap in how we interpret these types of things. So this guy says, employee 479 doesn't have shallow breathing. You can give that one some more work. And so, the idea that like maybe this increased monitoring is going to bleed into our workplace or something like that, I hope it doesn't. But you guys are more than just a number, but there's a lot of, again, pop science out there about wearable devices, biosensor, and I think that telomeres and cortisol, as I tried to highlight, are part of this kind of quantified self-movement. So just some food for thought. But with that, I'd like to close by thanking my wonderful mentor, Mark Laudenslager, Gene Kuttner, on palliative care and aging research, T32 that I was funded on, and my current division of blood cancers and bone marrow transplant that's been generous to keep supporting me through this work. And finally, I just have to highlight some clinical initiatives. If you know any caregivers, I do a lot of work in this area, not just in stress physiology, but trying to bring more support to our caregivers. And so this is a support group I run. And then just finally, there's my stock photo of the mountains, my email address, the one time that we went to the bar, and the mountains in Vancouver. So there you go. Thanks.
Well, thank you, Tim, for that engaging talk. We have about 15 minutes for questions, so I'd like to open up the floor. We don't have a walking mic today, so please just speak up. I've been told that works fine in this room. Just use your loud voice to ask Tim your questions.
Okay. Any questions? I could answer this page. It probably is important. No, I'm just kidding. All right. Well, thanks everybody for coming.